speaker is Brent Horton from Millersville University.
So I work with Brant Ryder on wire-tailed mannequins. I'm relatively new to the mannequin world. He sucked me in when we were postdocs together many years ago, and I brought with me this interest in endocrinology and how to integrate that and better understand physiological basis of behavioral ecology. And next thing I knew, he had me at Tucutini.
It was the opposite way around.
Down there sampling birds, and I've been hooked ever since. And we, one of the things that we found in our initial collaboration together is that we found that the highest social level for males, the territorial definitive plumage males, have higher testosterone levels than all other social classes. And same thing that Emily found in her work when she looked at testosterone, and we've seen, some other things. So testosterone is very interesting to us. One of the reasons that we're interested in this high level of testosterone is as you know, these wire-tailed mannequins are cooperative display species, and high testosterone in these birds is not surprising from the sense that these are the breeding males. And these are, you'd expect to see high testosterone levels associated with high levels of display rate and things like that. But what you might not expect in species with high testosterone levels is cooperation. And one of the hypotheses that may explain this is somewhere down the line, there's been a dissociation of testosterone control of some traits, maybe like aggression. So to better understand how testosterone may underlie behavioral phenotype in the species, particularly in the context of behavior in the social network and connectivity in the social network and things like that, we aim to sacrifice a number of males each year. Particularly, we want to focus on the definitive plumage class and territorial and floater males. And we want to harvest the brain and do region-specific gene expression analyses. We want to try to get a better idea of the neurogenetic correlates of variation in social behavior. And we want to take a region-specific approach because we want more, a little bit higher resolution than the whole brain approach. So we aim to look at multiple regions known as the social behavior nuclei, the lateral hypothalamus, medial preoptic area, things like that that are thought to be involved in social behavior. And we have a number of target genes in mind that we want to quantify and look for variation in expression, either with behavior or between social classes. And these involve, obviously, enzymes and receptors that lie along the testosterone signaling pathway. We know that that peripheral levels of testosterone are only part of the story, so we hope to look at the expression of genes in different parts of the brain, such as aromatase, 5-alpha reductase. We also want to look at the androgen receptor, as Barney and Laney have shown is very important, estrogen receptors. But we also want to consider some neuropeptides that testosterone could influence as well, such as vasotocin, mesotocin, and BMP. So my time's up. Thank you.